Did I get it?
You did. Very well done. Welcome! Today I have the ambitious goal of covering all the main, most important topics of chapter 15. And they start out, or we will start out with sex chromosomes and how problems are worked a little differently when the gene in question is on a sex chromosome. But let's step back just briefly, and I want you to tell me a couple things. How many chromosomes does a typical diploid human cell have? How many? 46. Because we have 23 pairs. 23 from one parent, 23 from the other parent, gives us 46 chromosomes. Now, only in meiosis do we actually make cells, gametes, or sex cells, sperm and egg cells, where that number is cut in half to 23, and those cells we call haploid cells. Now, you have genes on every single one of your 23 chromosomes and the 23 matching chromosomes in your cells. So you have 2 of each gene, typically. And out of your 46 chromosomes that you have in every single one of your diploid cells, so every one of your diploid cells has 46 chromosomes, 2 of them are considered your sex chromosomes, and they are paired up. They are a matching pair, or maybe not so matching. So only 2 of your 46 chromosomes are your sex chromosomes. And typically it's either XX for females and XY for males, are what names we give them. So that means you have 44 chromosomes that are not sex chromosomes. And we have a special name for them that you might want to note, but your book goes into this. They are called autosomes. A-U-T-O, like automobile, autosomes. That word autosome means a chromosome that's not a sex chromosome. And that means you have 44 autosomes. But there are 2 chromosomes that are your sex chromosomes. So your 44 autosomes and your 2 sex chromosomes add up to 46 total chromosomes. All right? Now, everything we've done so far, we have made the assumption that the gene is on an autosome. That it's an autosomal gene. So every time I've said like green and yellow peas, or snapdragons that are red or white, or that your blood type, you might have an A gene, B gene, or an O gene in some combination. All of that has been with the assumption that that gene falls on an autosome. So today we're going to talk about what happens when that gene is found in a sex chromosome. So there's 2 sex chromosomes in humans. There's the X chromosome and the Y chromosome. If you have an X and a Y, what gender are you? You're a male. Okay, so you would be considered a male when you have an X and paired up with a Y. Look at the difference between the X and the Y chromosome. That's the Y right there. It's a little stubby chromosome that doesn't do very much. Basically, this Y chromosome is a chromosome that has the information needed to make a person male. And that's about it. The X chromosome has much more information, and everybody's got at least one. If you've got 2 X chromosomes, that means you develop as a female. So instead of an X paired with a Y, it would be an X paired with another X, just like this. By the way, this is an electron micrograph, a picture from an electron microscope of the X and Y chromosomes. And this is at a stage where you have what? What are these 2 identical copies stuck to each other called? Remember that name?
Chromatids.
These are the sister chromatids. Very good. So usually you would just have one, like half here, but they are easier to see in this because they are literally twice as big, but it's only because this is a copy of this, and they're stuck together at the center. And you can kind of see that here with a Y, but it's not very clear. By the way, when I say that chromosomes get shorter and thicker, like in prophase, this is what we mean. They would be much longer and thinner and unfurled in, say, interphase. All right. Now, again, there are genes. This is all DNA in here, all wrapped up. And there are stretches of DNA all throughout here that control certain traits, and those are what we call genes. So different genes for different traits exist all along this chromosome and the Y chromosome as well. Now, in terms of how sex chromosomes work, in humans, and probably in many other mammals, I don't know exactly how sex determination works outside of humans for a lot of species. Humans have this XY system, as they call it, where you have, again, 44 autosomes, and then either an X and a Y or 2 X's to make you male or female. When you have only X chromosomes, your egg cells are going to only hold one X chromosome or the other at random. In males, since you have an X and a Y chromosome paired up, your sperm cells are either going to have at random an X or a Y. And what does that actually determine about your children? If your children are male or female, that is the father's responsibility here. Or, I don't want to say responsibility, but the father is the determinant of the child's gender. So fathers can give either X or Y. If they give an X, they have a daughter. If they give a Y in that sperm cell that's used in fertilization, then it will be a son. The mother always gives an X. So all of us, and this will come into play in just a second, if you are a man, you probably only have one X chromosome, and it was from your mother. If you're a man, you got your father's Y chromosome. By the way, your father got his Y chromosome from his father, who got it from his father, who got it from his father. So you can actually do genetic tests on a man's Y chromosome and genetically trace your father's 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 father. And we've done this and tried to look back in time genetically about 200,000 years. And there's a lot of interesting research out there on those kinds of topics. Now there are other systems, like some insects have this, you either have 1 or 2 X chromosomes that make you male or female. In chickens, you are male if you have matching chromosomes, and if you have 1 of each sex chromosome, that's what makes you a female. So it's kind of the opposite of the human system. And then here we have other insects, like in the bees, where it's so weird. I don't even know how this goes about working biologically. But the males are actually just the haploid organism, and the females are the diploid one. So there aren't really sex chromosomes per se, which is really, really unusual. So anyway, like with everything in genetics, usually it starts to get more complicated when you start looking at a bigger picture. And so just because something's true in pea plants doesn't mean it's true in humans. And just because something's true in humans doesn't mean it's true in some other animal or plant even. So here is what the genetics problems look like. Now, at first you might think, oh, this is scary. Oh, this is going to be too hard. It's not bad. It's not bad. Okay? This is a 2x2 Punnett square. It really doesn't work any differently than any of the monohybrid, simpler 2x2 Punnett square problems we've had. Okay? This is really no different from big Y being crossed with big Y, little y. But you do have to be a little bit more specific. So there is a trait called hemophilia. It is a blood clotting disorder that is recessive. You would know if you have it. There are a number of health complications that can arise if you cannot have your blood properly clot. For instance, if you were to have an injury, either an external cut or some sort of internal injury, you would bleed or bruise much more profusely than a person that does not have hemophilia. I'm going to assume all of us in here, I don't have hemophilia, and you would probably know if you did, but you probably don't either. It's a fairly rare condition. But here's the deal. The gene that controls whether you have normal clotting blood or have hemophilia is on the X chromosome, not on the Y, and not on any of the 44 autosomal chromosomes that you have. So when you deal with hemophilia genetics problems, and in this case they basically use big N to represent the dominant normal and little or lowercase n to represent the recessive hemophilia allele. And look at how this starts to work out. It's only on the X. So if you have a genetics problem, the female in the mating pair is going to have 2 genes because she has 2 X chromosomes. So in this case, we have what we would call a homozygous normal, or homozygous dominant, you could say it that way too, female. But you write out her 2 X chromosomes, and then as a kind of superscript, you note what alleles she has on each one. All right, so again, unless you're a girl in here whose father is a hemophiliac or whose mother was a carrier, and we can talk about all those terms in a second, most of the girls in here are probably X, big N, X, big N, which just means homozygous normal, homozygous dominant. This is how you would write the male, X and Y. And in this case, we have a male that would have hemophilia because on his only X chromosome, he has the affected gene, or the allele that causes the disease. Notice there is no superscript on the Y. You don't write the allele on the Y here because it's not on the Y chromosome. So this is where it gets tricky for guys. If a gene is on the X chromosome, and in humans, you're a man, and you only have 1 X chromosome, you only have one gene, and whatever gene that is, it's showing up. It's making the calls. So hemophilia is actually much more common in males because you either have it or you don't. Whereas in females, you might be a carrier that doesn't suffer from the condition. And we'll see an example of that here in a second. By the way, if we call this homozygous, and if you say, or a female that has a normal and an affected gene, we call this heterozygous. There is a name we give for what the male condition is, which is not two of a gene, but just one. We say hemizygous. You might see that word come up from time to time in the reading. So this is a hemizygous, one gene, or half the number of genes that you would expect. And that's the case with all males. So if you're a male, you either have the normal gene or the hemophilia gene.
But so what's the definition of hemozygous?
Hemizygous, H-E-M-I. Good question. Hemizygous means you only have one gene instead of the two you would normally have. So you only have one gene when you're hemizygous. Now check out the Punnett square. It's easy. I promise you. Father, possible contributions from the father are written on the top. Possible contributions from the mother written down the side. This woman here can only give an X chromosome with the normal gene. Both her X chromosomes carry the normal gene. So she's either giving one X chromosome with the normal gene or the other. The man gives, as always, either his X or Y chromosome. But if he has hemophilia or one of the other X chromosome linked traits, giving an X chromosome for him means giving the recessive gene that causes this disorder. So when you do the Punnett square, by the way, ignoring hemophilia or whatever the trait is, what are the odds you would have a boy versus a girl if you fill out this Punnett square? It's 50-50. Yeah. This is why we're about 50-50 male-female in the human population, is because any cross between a woman and a man is going to result in 2 of 4 being XX and 2 of 4 being XY. So it's 50-50. Now, if you have a man with a recessive X-linked condition, he's given it to his daughters. But their normal mother will give them a normal gene, and they will be what we call carriers, or heterozygous, because they have a dominant and a recessive gene. Women with this genotype do not suffer from hemophilia. Their blood clots normally. When they get a cut, it heals normally instead of just continuously bleeding. Notice, what are all the possible boys born from this mating? What's their situation? They're all normal. Every boy born to this couple will be normal for this condition because it's only on the X. And where do they get their X? They're completely normal mom. Good. So here's another possibility. Let's take one of these female carriers. Here we have a woman. She doesn't have hemophilia, but she carries the gene for it right here. And let's say that she has children with a man that does not have hemophilia, which is probably like all of the guys in here, unless you know you have hemophilia. In this case, things start getting flipped around a little bit because this mother can give either the normal or the abnormal X chromosome that she's carrying, and the father will either give you a normal X chromosome or a Y chromosome that doesn't carry the gene at all. So now that the mother, in this case, actually carries this affected gene, we start seeing her sons get it. And this is a pattern you will see with sex-linked traits. Mother carries it. How many of her sons? What proportion of her sons could get this?
1/2.
A half. Every son will either get the normal or the abnormal gene. So you start seeing carrier mother with a father that suffers from the condition. If he has daughters, what's he given his daughter?
The gene.
The X chromosome with this affected gene. So if you have a man with hemophilia, all his daughters will be at least carriers. And then if they have a son, there's a coin toss that one of their sons will be affected. This kind of male-female distribution does not come up in autosomal genes. In the 44 chromosomes, all those genes, you would just write it big A, little a. But if you have genes on the X chromosome, and I would have to tell you this is a sex-linked trait, then you need to start writing out the sex chromosomes and including them in your Punnett square. Again, mother is a carrier. Half of her daughters will be carriers as well because she has a 50-50 chance of giving that affected X chromosome. And then we have this situation, which would be pretty rare, but this happens, where you have a man with hemophilia. And by the way, red-green color blindness works this way, and a few other traits, but hemophilia is kind of the one we always discuss, typically for whatever reason. But in this case, we have a man with hemophilia and a woman carries it. Notice there is a chance to have a girl with hemophilia. This is the only way you would get this, though, is to have mother as a carrier and an affected father. So women typically are protected from certain sex-linked conditions because they have a good chance of actually having a normal dominant allele as one of their two. But with guys, they don't have another X chromosome that might cover up this disorder. So as soon as they get the gene, they got it. Again, I can't stress enough, in all three of these examples are simple Punnett squares. You just have to make sure that you're including the X's and the Y's, and that if it's on the X chromosome, you don't include that letter on the Y chromosome. You don't represent that gene on the Y chromosome. So, let me ask you a few quick questions then. If you have a completely normal mother when it comes to hemophilia, can her sons get hemophilia?
No.
No. Even if their father has it, her sons are completely safe from this condition. And oddly, that's kind of like a mother-son connection. Oddly, it's the father, if he has hemophilia, that is passing it on, but only to his daughters. And all his daughters are going to be carriers for it because to have a daughter, he has to give his only X chromosome, and it's got the gene. So you start seeing these gender connections from parent to child. Okay, we're going to practice some problems like that. The next few slides might start going over your head a little bit, but only because primarily there are some really weird symbols that you might not follow along with. So I would really encourage you for about the next 5 or 10 minutes, try to really focus on this. And then the last few slides are pretty straightforward. We actually go back to meiosis and talk about what can happen when things don't go according to plan. So this is an experiment from flies that was done about 100 years ago now, in which things start out looking pretty normal, but then at the end you start seeing some weirdness that we really haven't talked much about, but I've mentioned it. So we're dealing with two traits here in these flies. The color of the fly's body as gray or black, with it being dominant as gray and the black as recessive. And then these flies can also have what we call normal wings, or these not really functioning wings caused by a condition called vestigial wings. Again, dominant is the normal condition, and this vestigial wing is the recessive. All right, now where it gets a little bit weird is that the dominant gray body color is given the symbol typically, and your book uses this symbol so it makes it look funny, B+. And the recessive black body is B without the plus sign. It's kind of weird, But you can use whatever symbol you want. This is the symbol that the geneticist that did this experiment originally went with, and these are symbols used a lot in genetics. The normal wings are given this VG plus symbol. I really wish they just would have gone with like big N for normal, but they didn't. And then these weird non-functioning wings are the VG symbol without the plus sign. So basically you have these are the two chromosomes that this individual has, and we actually know where the genes are on these chromosomes, and this individual is homozygous dominant for both. All dominant genes. 2 dominant gray body genes, 2 dominant normal wing genes. They mated these flies with males that were homozygous recessive for both traits. We've talked about this. We've seen this already. When you do this, when you mate homozygous dominant with homozygous recessive, all of the offspring have the dominant appearance. So all of the F1 flies have the dominant gray body, the dominant normal wings. Nothing's weird here. Exactly what we expect. Now, what do we call this condition where you have a dominant with a recessive? This is heterozygous. So this offspring, while looking dominant for both traits, is actually heterozygous for both traits. Okay, we've been here before. Purple, dominant purple, recessive white flowers. We got all purple flowers. Same thing, except we're doing 2 traits at once. Now, this is where we discovered crossing over is occurring. Because what they did, and I probably should have actually shown these slides out of order to make them make more sense, is in this experiment, they took this heterozygous F1 offspring and mated it with a recessive fly again. Completely recessive male. Now, here's what they expected. They expected that you would have about 50-50 dominant genes from the dominant parent matching up with the recessive genes from the recessive parent. And since the parent was heterozygous, their recessive genes matching up with the recessive genes of the recessive male. But what they ended up finding was totally new combinations they had never seen before. They started finding flies that were actually heterozygous for one of the genes, but homozygous for the other, and vice versa. They started finding weird combinations, and those weird combinations are called recombinant offspring or recombinant genotypes. And to go back to the previous slide, here's what was happening. In prophase 1, we have what's called crossing over, where pieces of homologous chromosomes can break off and switch places. So what can happen when genes are on the same chromosome is, and you can look right here, you have one chromosome that's completely dominant, one chromosome that's completely recessive. It's heterozygous. But during prophase 1, pieces of these chromatids can break off and switch places. So now you've got what should be a copy of this chromosome is mostly a copy with a piece of the pair switched. So you took a piece from this chromosome, stuck it to the end of here, and that piece you broke off here gets put on the other one. So you get these unusual chromosomes that the parent doesn't even have. This parent had a chromosome with 2 dominant genes, a chromosome with 2 recessive genes, and now they're making chromosomes that have 1 dominant, 1 recessive. We have recombined the chromosomes. And if they are used in the gametes for fertilization, then you get individuals that have these recombinant chromosomes. And you start getting phenotypes that you would never expect to get. Now, we can measure how common this is, and it actually tells us something that I'm going to point out on the next slide. So in this experiment, they finally figured out what was happening was this. As I just showed you, between the body color gene and the wing shape gene, the chromosome was breaking off and switching places with another chromosome. And they figured that this was happening in this specific case with black and gray body color and normal or vestigial wings about 17% of the time. And then they started finding other traits that this was happening with when you had breeding occur in these experiments. And what they started doing was mapping where these genes were on the chromosome based on how often they were separated from one another during crossing over. So the black-gray body color genes and the shape of the wing genes, since they were breaking away from each other and recombining about 17% of the time, they knew they were further apart than when they did other experiments and looked at other genes that only broke off and switched places 9% of the time. So they're able to figure out that the more often genes were reshuffled during crossing over, the further apart they were, and they were able to mathematically map out where the genes were on chromosomes. So they started taking this genetic experiment, or these genetic experiments, and figuring out where along the chromosome and the distance between each other these different genes were. And so they started doing it with like these little antennae and eye color and the body color and all of that. So they're able to start mapping where the genes are on the chromosome based on how often crossing over broke genes apart from each other during meiosis. It's not easy reading. I don't expect that you just understood everything I just said. We will not be doing any gene mapping based on recombinant frequencies in here on the test. I'm not going to give you practice problems with it. I would doubt it would show up on the AP class or on the AP exam at the end of the year. And I couldn't imagine throwing this mathematics at you in this unit along with all this other stuff and still staying on track. But I do want you to understand that if you have two genes that are really far apart, there's a lot of space between them where they could break during crossing over and switch places. But if you have two genes that are very close together, there's less space between them where you would find a random break for crossing over to occur. So genes that are far apart but on the same chromosome, they are more often shuffled in the deck. All right? Try to follow along with that in the reading when you do it this weekend. It's not easy, and I want you to understand it on a more general level. You don't have to come up with actual like units of distance on a chromosome between two genes. I'm not going to make us do all that. Okay, finally, and this is where we're going to kind of wrap this chapter up. Chromosomal disorders. You're probably somewhat familiar with this concept. This slide shows 4 major ways in which a chromosome can be altered in a way that makes it an abnormal chromosome. And usually there are symptoms or phenotypes that arise. And these are not, these are genetic, but they're not caused by genes. They're caused by an entire chromosomal problem. So the letters here represent different genes along a chromosome. Sometimes an entire gene, a whole section of chromosome, can just be deleted. We call it a deletion. Anytime you lose DNA, we call it a deletion. Sometimes it's a single letter in a gene. Sometimes it's the whole darn gene. A whole section of chromosome can be deleted, and the chromosome will actually be shorter than it should be. You can see this when you look at a person's chromosomes. We can actually take cells out of an unborn baby, look at the chromosomes in that cell and say, this baby will be born with a chromosome that has a deletion. And we might even be able to predict what would result from that deletion. There's what we call duplication, where you can actually take multiple genes and duplicate them, copy them, and then you have a chromosome that's bigger than it normally is. That's pretty self-explanatory. There are weird things like inversions, where the order of the genes can get out of place, which can cause problems. And then translocations, which are probably the easiest ones to see, are when a piece of one chromosome switches places with a piece from another chromosome, but they're not matching homologous pairs. It's okay if you switch this A and this A, the baby will be born with gene A. But if you start switching the entire chromosome that it's on, those genes may never be read because they're not appearing on the right chromosomes. Or they may be read too often. And that can be just as much of a problem as losing a gene. And that's called translocation, where a piece of one chromosome breaks off and attaches to a piece on another chromosome. And you can actually sometimes see it in a picture of chromosomes. You can see a little chunk on one chromosome that should be on another. So how can things like this happen? How can you get certain chromosomal disorders? Well, one way you can get chromosomal disorders is through a process called non-disjunction. And this is the last term I'm going to tell you about. The next slide is just a picture of the karyotype. During anaphase, what happens? What's a big event during anaphase? The chromosomes pull apart from each other. That's right. So, in the case of anaphase 1, these two pairs, these matching pairs, should be pulling apart, one pair going to each side. But sometimes the pairs stick together. And if that happens, you end up with gametes, sperm cells and egg cells, that might have one more or one fewer chromosome than the cell is supposed to have. And if that sperm cell with an extra chromosome, or that egg cell with an extra chromosome, or that sperm or egg cell that's missing a chromosome, if it's the gamete used in fertilization, the person resulting from that sexual reproduction is not going to have a chromosome or will have one chromosome too many. And you see that in Down syndrome, which is the most common example of this. Notice here we have two X chromosomes. What does that mean?
Girl.
It's a girl. But for chromosome 21, we have 3 homologous chromosomes instead of the 2. There is too much genetic information, and it causes kind of an interference signal, and there are certain developmental complications that result from this. This is called trisomy. 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 Trisomy is when you have 3 of a chromosome instead of 2. If we call 3 trisomy, what do you think we call it when you are missing a chromosome and only have 1 instead of 2? It's monosomy. Very good. Like a monosomy.
That was close.
Excellent. It's just the syllable he was saying. Monosomy or monosomy. You could say it like that. By the way, monosomy, when you're missing a chromosome, is fatal. Or at least very, very impactful. There's very few chromosomes you can live with only having 1. The one time that I know of that it is not fatal to be missing a chromosome is, what's the least useful chromosome, ladies? What chromosome is small and stubby and not very useful?
Why?
The Y. You can live without a Y, and there are people born with 1 X and nothing matching.
Oh wait.
It's called Turner syndrome.
Is that one like they say like you're a girl but really?
No. What gender are you if you have X nothing?
Girl.
You're a female. Now, they typically don't sexually mature quite properly, so I think they're not fertile individuals, like they may not be able to have children of their own. But missing a Y and just having a single X chromosome is the only case of monosomy that I know of in humans that you could actually, like, that the baby could even be born, could make it that long. By the way, there are thousands of these mistakes that can be made in humans. You can have XXY. You have a Y chromosome. You have male genitalia, but you have an extra X chromosome. There's XYY. It's called super male syndrome. Doesn't really seem to have any effect. You have super female syndrome, which is 3 X chromosomes. There are some issues there. The X chromosome carries a lot of information. Having an extra one seems to be somewhat problematic. You can have trisomy 21, which is Down syndrome, but you can also have trisomy 20, trisomy 19, trisomy 18, trisomy 17. You can have all sorts of trisomies. You can have an extra of any chromosome, all because of non-disjunction. The chromosomes don't pull apart properly during either anaphase 1 or anaphase 2. Okay. So those are the highlights of chapter 15. I know that took quite a while, but we are done with the new information. It will be on you this weekend to go through chapter 15 on your own as much as you can and come talk to me about parts you don't understand. But that's all the class time we're going to spend on it because there's a lot of practice problems and test preparation I want to do next week. So you can stop the video. Make sure you try your practice problems online.